So I'm Olivia Goldring, and I worked at the Brain Research Lab in the Psychiatry Department of NYU under the leadership of Dr. Leslie Pritchett. So I worked in a clinical study that used EEG brain imaging to quantify and localize pain in the brain and further understand the encoding of pain and where specifically it occurs in the brain. Okay, so an EEG is a neuroimaging method that measures neuronal activity in terms of frequency. So each brainwave has a specific frequency, and electrical activity in the brain can be analyzed in order to understand many of the functions of the brain. So the pain matrix is a network of structures that are connected in encoding pain in the brain. So the goals of the study were to use EEGs in order to localize pain, quantify pain, and identify activation of the pain matrix. So here we can see these four images. This is an image of the lateral view of the brain, and you can see the structures in color are those cortical and subcortical structures that are involved in the coding of pain in this pain matrix, and the same for the picture right there. So we can see, so we saw that in this study, all the patients in the study experienced neuropathic pain, which is chronic pain resulting from injury to the nervous system. So many had pain from past spinal cord injuries and others from chronic migraines. So they were all uniform in the fact that it was neuropathic. So using Loretta and Beretta, these are two different methods. Loretta is low-res electromagnetic tomographic analysis, which is just a method that allows one to see the total electrical activity in 3D. So it creates a model using the EEG in order to see the different brainwaves which differ in frequency. So the quantitative EEG may be a means of detecting pain in patients who experience chronic pain, and we found that EEG is actually clinically practical in comparison to other neuroimaging methods such as MRIs, PET scans, and fMRIs. So here you can see that there are clear differences in these 6 patients. So there's high activation in the bright red and yellow. It was seen in the high pain state in the regions of interest of the pain matrix, and there are significant decreases in activation in the low pain state, which is seen in gray and less color. So EEG recorded data of all 50, so this is what EEG, the raw data actually looks like. So we see that the brainwaves emitted from the brain are measured by electrodes placed on the scalp, and this is just what it looks like. And from here we use this to simulate models, 3D models, and this is for red. These are all kind of Retta images. You can see the different hertz levels. So yeah, these Retta images on the right display the 3D models of the total electrical activity taken from the EEG raw data. So they allowed us to see exactly where in the brain the electrical activity is enhanced, and it allows us to localize and quantify the pain, and reiterates the specific structures that are involved in the pain matrix that encode pain. So we saw that the EEG of a person experiencing chronic neuropathic pain compared to that of a normal person is extremely different. Side note, I was actually a control in this experiment because I don't experience neuropathic pain. So you can just see that like my EEG is significantly different than that of someone who has past spinal cord injury or other types like migraines or something like that. So as you can see right here, that's what an EEG looks like. Not the EEG data, but that's basically how an EEG is administered. So you have these electrodes placed all over your scalp, and it measures the electrical waves from the scalp. So we saw significant differences between chronic pain patients and the normal controls, and there's a big difference in the high and low pain states, and EEGs can be used to quantify and localize pain. So as you can see here, in patients with high and low pain states, the pain scores right here are seen, they were taken from these different questionnaires and self-administered different things that we gave to the patients. So you see that in red, high red and yellow, you can see in the high pain state there's a huge difference from that in the low pain state, but you see that in the low pain state it's the exact same structures that are involved in encoding the pain. Thank you.